All right, in 25.4, we're going to talk about a different way of identifying microbes using immunological methods, which means using basically antibodies to identify specific antigens. So we're going to talk about the enzyme-linked immunosorbent assay, the Elisa. We've talked previously a bit about that in the immune system chapter, but we're going to go into how we can actually apply that in the clinic, and we'll also talk a little bit about fluorescent antibody staining. So all of this is using immunological tests, and we can tell if there is a current infection. We can also use this, interestingly, to find evidence of a previous recent infection when we look at patients' antibody levels. So let's look at a case study here. In March 2014, about 60 people in the southeastern forest of Guinea in Africa became ill. They had high fever, diarrhea, headache, vomiting, gastrointestinal bleeding that comes out the rectum. At least 23 of those people died, and based on these symptoms and the location, the presumptive diagnosis is Ebola hemorrhagic fever, one of the scariest diseases out there, about a 50% chance of dying if you get it. We have to confirm this, so they send samples to the lab, and we're going to use different techniques to identify this. So we're going to use viral antigen identification tests. So we're going to use an ELISA that is going to recognize the viral antigens, and this is a great way to identify viruses. In this case, they confirmed it. They also tested whether the patients were making antibodies against Ebola. This uses a slightly different test, but in that case, again, they were positive. So once it's fully identified, you implement super strong quarantine mechanisms for Ebola. They hoped to prevent this outbreak from growing to other villages and countries. Unfortunately, by August of that year, the epidemic had spread to other countries, Sierra Leone, Liberia, and to Nigeria through air travel. So by then, there were about 1,300 presumed or confirmed cases. And again, half of those people died. So at this point, they're very worried about it spreading out from Africa to other regions. So by January 2015, 9 months later, there were over 7,000 deaths from this. So luckily, we got a handle on this, and it did not continue to grow too much larger. But Ebola is one of those things that we worry about quite a lot because of the high death rate from it. So rapid identification is key. That 50% death rate requires us to instantly test people when we suspect this. And to do that, there is a test kit. This rapid test kit here is an ELISA test kit that finds and binds to the Ebola virus glycoprotein, which is one of the antigens on the surface of the virus. So this is identifying viral antigens, and this is super quick. This is even quicker than PCR in a lot of cases. So you'll remember the enzyme-linked immunosorbent assay, the ELISA. This detects either antibodies or antigens, and it uses antibodies to do that detection. There are several ways we can test this. In the previous case that I just showed you, we used antigen capture, where we have an antibody that binds to the Ebola antigen, and then we have a secondary antibody that comes in and makes a sandwich, and this causes a reaction to occur which changes color. And we can actually use a machine to read that color and quantify it, so we can actually automate this process a little bit. In the case of Ebola, this works very well early in the disease when human antibody levels against Ebola are low. We can also use a different method called antibody capture, where we test patients' blood and we find antibodies against Ebola in there. So we're actually using an antibody to bind an antibody. It's a bit Inception right now, right? But this works well later in the disease when there are high antibody levels in there. So this uses a slightly different test. We bind Ebola antigen to the side of the tube, and then we apply human serum. If the antibodies are in there, they will bind to it. Then we can add a second antibody that will bind to those ones and give us a color. So let's watch an animation of this in action.
An enzyme-linked immunosorbent assay, or ELISA, is a way of detecting the presence of an antigen by using an antibody-conjugated enzyme to cause a color reaction. The sample is immobilized by direct adsorption to the well of a microtiter dish. If the correct antigen is present in the sample, the antibody can bind to it. A washing step removes any unbound antibodies. Now, adding the substrate results in a color change reaction catalyzed by the enzyme. The intensity of the color change reflects the amount of the antigen that bound to the well, and thus was present in the patient's sample. This example shows the direct form of the immunoassay procedure, in which the tester antibody is directly binding to antigens from a sample. In another form, called an indirect assay, serum antibodies are removed from a patient with an infection, and these antibodies are tested. Here the question is whether the patient has a hepatitis virus, so a known hepatitis antigen is fixed to the well of a microtiter plate. If the patient's immune system has produced antibodies against this antigen, they will bind. A washing step removes unbound antibodies. Now a secondary antibody, conjugated to an enzyme, is added. The secondary antibody is specific for human IgG isotype antibodies. A washing step removes unbound antibodies. Adding the substrate results in a positive color reaction. Another indirect form of ELISA, called a capture assay sandwich, begins by binding what is called a capture antibody directly to the microtiter well. The capture antibody is specific for a given antigen. Here a patient sample is added and the antigen of interest is captured. Now an antibody to detect the antigen is added, followed by a wash step, followed by an enzyme-conjugated antibody that can detect the previous antibody, followed by a wash step. The development of color identifies a positive reaction. An indirect ELISA is important in identifying patients with Ebola. This microtiter assay plate has 96 wells, which allows testing multiple patient serum samples simultaneously. The wells contain known Ebola antigens attached to the surface. A person with Ebola will produce Ebola-specific antibodies. The Ebola-specific antibodies, if present in a patient's serum, will bind to plate-bound antigen. A washing step removes unbound antibodies. A second antibody, conjugated to an enzyme, is added. This antibody binds to human IgG antibodies. This particular antibody comes from a rabbit, so it will not bind to itself. Washing removes unbound antibodies. Adding a substrate results in a color reaction in wells that have retained the enzyme-bound antibody. The rate of conversion of the substrate is proportional to the amount of anti-Ebola antibody present in the patient's serum. An early laboratory diagnosis of Ebola using ELISA can lead to quick implementation of supportive measures that can improve chances of survival.
So the key thing about ELISAs is that they're super fast. They can work very, very quickly. So we can use them to quite rapidly identify either antigens, so the indication of the pathogen, or antibodies against that pathogen. We will see this being used in the next section. I want to finish up with antibodies when we're talking about fluorescent antibody testing. We can make specific antibodies that recognize specific bacterial species. Here you can see one that recognizes diplococci, so pairs of cocci. Here we're recognizing rod-shaped bacteria. This can be very useful to identify a suspected microbe rapidly, because all we got to do is take like a blood sample or a skin scraping or a swab or something like that, do a streak, and apply these antibodies to a microscope slide. And we can then see this reaction very, very quickly, positively identifying a microbe. So there's always the question of what tests to use when. Obviously, easily grown bacteria can be cultured, and this is especially true if it's not time sensitive. It's much cheaper, and if it's not time sensitive, we don't worry about that too much. Biochemical tests can be done, and we can use some automation in that process. Things that are difficult to grow, particularly viruses, we often use nucleic acid or DNA tests, or those immunological antibody tests to identify them. PCR is a little more sensitive than the antibody tests, so to detect very small amounts, PCR is the best, and we can actually quantify very well using PCR. Microscopes are still super useful, right? Particularly for eukaryotic parasites. They're very large, they're very easy to identify. You can't culture them on plates. You could do PCR, but we don't suspect them as our first thing there. So microscopes work well for eukaryotic parasites. I've included some different diagnostics that are used for different diseases if you're interested in here. Things like Yersinia pestis, the plague, fluorescent antibodies, or Elisa are commonly used. PCR can be used for that as well. Rhinoviruses, the flu, all of these. For things like this, we're using qPCR a lot of times, or those antibody tests. We have qPCR for SARS-CoV-2 virus. There are also rapid antibody tests that we can do. You can test for the antigen, or you can test people for the antibody, although that just shows that they've had SARS-CoV-2 or are vaccinated against it. All right, so we talked about the use of ELISAs to either detect antibodies that the patient is making in their serum or antigens from a virus or bacteria or something like that that are in the serum there as well. And we can even use fluorescent antibodies to quickly identify things on a microscope slide. All right, that's it for 25.4.